All right, so it's my absolute pleasure to introduce Fiona. Many of you know that I did my PhD with Fiona, and in my second year of PhD, her son was born, and she was on the phone with her son and just casually mentioned he's 12 years old, so that's daunting news for me, that's for sure. So Fiona really doesn't need a lot of introduction, and I do encourage, she provided a really comprehensive bio on her for this talk, and so I do encourage you, if you want to find out more about what Fiona's professional achievement, to go and check out on our website what she's been doing. But I just want to mention a few things. So she's a professor in molecular biology and biochemistry, and an associate professor in School of Computing Science and Faculty of Health Sciences at SFU. And she's the coordinator of a large consortium of a large genomic database consortium involving researchers from 13 different countries, and leading multiple projects in spanning microbial genomics, metagenomics, public health, microbiology, and so on and so forth. Large number of collaborators I won't mention one by one. She is on several committees and boards, notably including as a board of director on Genome Canada and scientific advisor board for the European Institute, European Bioinformatics Institute, EBI. Okay. And she's also mentioned that she's passionate about mentoring, and I can certainly attest to that from my first-hand experience. And she has numerous awards, including Canadian Society for Microbiology Fishers Award. A lot of these I knew about, but I better read them just so I don't get them wrong. And so I remember when I first joined, she was awarded as a top 100 under 40, I believe, at the time. 40. Top 40 under 40, okay. And also named as the 100 of the world's young innovators in science and technology. And she's most recently recognized as Thomson Reuters highly cited researcher, as the top 1% highly cited researcher in her field. So without further ado, give you Fiona. Her talk will be simply called Microbes.
Well, it is my absolute pleasure to be here. I have to say some of those awards are thanks to Will, who did graduate studies in my lab and did some phenomenal work. So I have to say kudos to him, and also I have to say a big thanks to the VanBug team, and just say that really I probably should have called this Bugs and Bugs for Bioinformatics Users Groups, because really what I'm trying to do is get people who are doing bioinformatics to get interested in microbiology and realize the power of what you can do in microbiology now, and why microbes are really cool and very important. First I'll just mention, I just have to do a little shout out to others who might be joining in, because this is our first time we are webcasting this, including groups that are hoping to form their own bugs. We've got Torbug in Toronto and Monbug in Montreal that already exist. And I was just in Calgary at a bioinformatics symposium where they were wanting to set up a CalBug. And a big shout out to Steph, who's interested in doing a CamBug in Kamloops. Yay, Kamloops. And I was trying to come up with colors, and I thought maybe like a nice sandy, sunny color. It's very sunny there. But I've got a little bug here, a little opening here for anybody else. Let's keep replicating these bugs and have some more. So first, I'm going to start with some crash course on microbes. I want to make sure that this audience, actually, I just want to check, how many of you would say you do microbiology research or are microbiologists? Can you just do a show of hands? Okay, so like maybe less than half, you know, so there's a lot of people who aren't here. So yay, I'm going to get your, twist your minds into thinking microbes are super cool. And, but first what I want to do is make sure you guys are all up on some basics. And first and not least is that microbes are not all germs. The vast majority of microbes are actually not disease causing, and that's important to remember. Oops. As you go forward, that microbes are basically play a very intricate role in the cycle of life, and they're not just about degradation. They play key roles in many components of many life cycles. And in fact, I'm going to do a little pop quiz here. Does anybody know roughly what percentage of oxygen on our planet is generated by microbes? Does anybody have an idea? Want to volunteer a number? What? 30%? That's a great guess, and I really honor that. Anybody else want to mention something higher? 60? That's good, yeah. So actually it is over 50%. So over 50%, you know, I've heard numbers like 70 and 80, but, you know, as you know, it's a bit of a getting an accurate number. But, you know, we're talking a very significant percentage of oxygen on our planet is generated by microbes, let alone other, you know, important roles they have. So I just want you to appreciate these are very, very important organisms on Earth. And not the least to say, there's all these fine foods that I certainly recommend that are all as a result of microbes, including fine beverages as well. And for those who aren't familiar with microbes, they come in all shapes and sizes. My favorite, I think, is these ones that look like wine bottles, to continue with the wine theme. These are archaea viruses. There's lots of really cool ones out there. And I'm going to do one more pop quiz of anybody know how, some of the microbiologists might know how big microbes are, but do you guys know what the lower limit of the resolution of the human eye is? And before you Google it, if you Google it, it actually gives it to you in pixels. So it's not actually, I want in millimeters. So how small do you think you can actually see? 0.1, good, not bad. So I mean, what I've heard is sort of 0.1 to 0.3 millimeters. I think I'm probably in the 0.3 millimeter because I'm getting older. But basically, you know, keep in mind that sort of if you can sort of see something that's sort of 0.1 to 0.3 millimeters, it's about 1,000 times smaller, you know, when you get to the smallest bacteria where you've got diameters of 0.2 micrometers, okay? And that's like getting to be like the smallest. Obviously, there's a lot that are in sort of the 0.5 micrometers and bigger, up until about the size of the resolution of your eye, hence why they're called microbes, because the idea is they're supposed to be too small to be able to see with the naked eye. So, you know, keep in mind these are very, very small. Because they're very small, there's a lot of them all around, and in fact, you know, one of the stats, there's many stats you can rhyme off about all the microbes, and so here's a nice one about the number of bacteria living in your mouth can easily exceed the number of people who live on Earth. So there's a lot of microbes there because they're very, very small and they're packed in, and you know, the famous stat is that you've got more microbes in your body than different microbes in your body or just microbes in your body, period, then you have cells, human cells in your body, so you're technically a little more microbe than you are a cell. You're really walking vessels of these microbes. But essentially, what I wanted you to appreciate also is that, you know, there's a lot of these microbes, and so we've tended to increasingly do a lot of microbiome analyses, and I have to say, I'm going to start off with, because there's been some debate about this recently, clarifying that I tend to use the words microbiome and microbiota interchangeably to imply a collection of microbes in an environment or sample, and then metagenomics is a study of the DNA required from an environment or sample, in keeping with a nice post by Jonathan Eisen who sort of addressed this. So just be aware that that's what I'm using as my terminology. But I just want getting back to all the good microbes out there. These good microbes aren't just in your body and doing things that are very useful in terms of helping you with digestion and helping you with metabolism, so that you end up getting some nutrients. They are also acting as an important defense against pathogens. Its early exposure to microbes has been increasingly shown to be very important and key for your immune system. And if there's one bit of advice I always give to new parents is make sure you get your kids out and just play in soil and play in dirt, because the best way to protect your child from allergy and asthma is considered to be to get a dog. And the reason for that, they think, is because the dog is literally tracking microbes in and out of your house, because they go in and out of your house so much, and that's actually giving your child exposure to microbes at a level that is actually more natural versus what we used to live like. And so you can sort of consider this not man's best friend, but child's best friend. And so if you want to see more, Jack Gilbert has a really nice little video about how dogs can be beneficial to your health thanks to bacteria. But the point is that you basically have all these good microbes out there, and so we published a little paper led by Kieran Doherty and Rob Biko about the human microbiome and sort of stewardship of that, and the concept of, and I would argue that it's not just for the human microbiome, but for just microbiomes in general, that we really should be starting to practice conservation efforts. We've tended to focus a lot on having these different conservation efforts for conserving our diversity of animals on Earth and maintaining them and monitoring them. We should also be doing the same for microbes, because we're starting to appreciate just how important they are, and maybe some of the conservation approaches could be applied to microbiomes as we start to interrogate them more. But do realize that microbes can obviously play a role in disease, and in fact, it's not just infectious disease, more complex diseases. Here's some classic experiments of transferring microbes from genetically obese mice to normal mice. Mice actually cause thinner mice to gain body fat. And my favorite is the jet-lagged mouse. So basically what you can do is take a mouse and you can give them jet lag by giving them a different light-dark cycle. And then you can, if you do fecal transplants from these jet-lag mice to germ-free mice, you can induce the same metabolic symptoms similar to jet-lagged mice in those mice that you don't get if you do the control of the unjet-lagged mice microbiome being put into the germ-free mice. So, and then what to me really blew me away was in this year by Leon et al., this microbially derived metabolites that they're suggesting modify circadian rhythms and host metabolic function. In other words, we've been tending to think of jet lag is something that's really important to watch, and something that we have all these metabolites that we have to adjust and there's a lot of interest in how we can reduce jet lag, but actually what I think is now being more appreciated is actually microbes play a major role in that, and actually it might be the microbes that are actually playing a predominant role in that. And so now I don't know about you, but I'm walking around with thinking these are my little babies that I got to look after, and I got to make sure I feed them, because apparently you have to, when you feed them is literally having an impact on when they produce these metabolites. And so just realize you've got literally like an organ in there of these microbes in your gut that you do have to take care of, and that play a very important role. Okay, so I also have to do a plug-in for Jennifer Gardy's awesome children's book about microbes. So if you want to know more about microbes, this is a fantastic book. This is a scientist at the BC Centre for Disease Control and also UBC professor who has a great career also in media. And this is a fantastic book, as endorsed by my 12-year-old son, who says it's awesome. You know, I'm sort of ending with going over this microbe and the importance of microbes. Infectious diseases obviously are still a concern. It's a small proportion of the microbes that are out there, but certainly there are infectious disease outbreaks that can have health impacts, and they can impact economies. You know, one great example was in 2011. There was an E. coli outbreak in Germany that basically they couldn't figure out if it was the cucumbers or the tomatoes or the sprouts associated with salads. They saw a lot of people getting sick eating salads, but they didn't know which component of the salad was. And because of the nature, the globalization of food production, it was really hard to track where this E. coli was coming from. And fortunately, a bunch of tomatoes sat rotting in fields where they falsely thought they were the source of the infection, when actually it was the sprouts through these Egyptian fenugreek seeds. And eventually this was figured out, but not before we had 53 deaths, you know, almost 4,000 people ill. And I think most notably, you know, we forget about the economic impact that these infectious disease outbreaks can have. I mean, this is, farmers were compensated for, you know, these, you know, tomatoes, et cetera, rotting in fields, but for 210 million euros. I mean, that's not trivial, right? So I think we have to appreciate that infectious diseases are still a notable issue, and particularly with our current global community and global travel, we do have to be aware that it's really important to sort of nip these outbreaks in the bud quickly. So, and then on top of that, we've got antimicrobial resistance is increasing, and this is a figure showing that basically 10 years after, about 10 years after any drug is developed, you end up getting resistance to those that antibiotic that's been newly developed. And so there's a real need to come up with new therapies, more sustainable approaches, what I call them, for dealing and controlling infectious diseases. So that's where I come in. My primary research interest is in, quote, developing more sustainable approaches for infectious disease control. And I'm interested in taking advantage of the fact that I was originally trained as a microbiologist, but I was always a computer geek, and so put those together, you become a bioinformaticist. And I'm interested in using novel computational tools, integrated data, and I really like using interdisciplinary approaches. So to do this, what I've been doing is looking at sort of the host, pathogen, environment, sort of triage of the triad, I guess I should say, of components of what leads to infectious disease. So you've got these pathogens that I'm interested in developing anti-infective drugs that don't actually kill the microbe, so they have less selection for resistance to develop. It's basically converting the microbe into a non-pathogen, or putting pressure on the microbe to basically potentially do that, allowing the immune system to come up and basically control things without damage occurring due to a toxin, for example, that you can target. I don't have time to talk about that component of it, but I will say we've had a lot of fun doing repurposing of existing drugs, including some, unfortunately for men, we found some really good anti-infectives that are basically endocrine, used to treat bone density issues in women because they're endocrine modulators that probably you wouldn't want to take because they might affect your, they're sort of estrogenic, but still we're working on it, and we've got some great structure function studies where we can see by using computational approaches where we're doing docking, we're able to basically identify some possible compounds, and then we're testing those in a C. elegans worm model of infection, and we're able to actually find some drugs that can protect against infection, but they don't actually, the drugs don't actually kill the microbe, okay? So then what I am going to talk more about is just some immune modulator development, just because this also involves looking at the host, and there's a bunch of you who don't study microbes here, so you might be interested in aspects of that research. And then also some work when we look at the environment. A lot of it is looking at the microbiome environment, and also using genomic epidemiology to track outbreaks and identify what sort of social or environmental factor played a role in an outbreak. And lastly, I'll just mention that these methods do have applications in environmental research, allergy research, agri-food. So I'm basically going to give you one little example of how we've used that. But before I get going with that, I just want to mention a little bit of research philosophy because, there's a lot of students in the room, and I want to make sure you appreciate that I've really benefited. I've gotten awards, but a lot of it is actually just because I just focus on just doing very quality, open research, remembering the big picture, and integrating ideas from other fields. You talk to people from other fields, and it can really get you to think in a different way and come up with truly new ideas. So I really encourage that concept of, branch out and look at other areas, talk to historians, talk to people in different areas of mathematics, etc. And you can get some really great ideas, talk to people doing different kinds of computer science, and then try to apply that to your particular bioinformatics problem. So the other thing was that I really like the idea of doing sort of getting robust data when I can, using very accurate analysis methods and really organizing data. I think that's the nexus of really doing very good quality bioinformatics analyses. The challenge is, sometimes it's hard to get that robust data, or you have to sort of accept the fact that that data is just not good enough, and you're going to have to get better data. But you can certainly, as a bioinformaticist, control the analysis methods you use usually and improve them. And then the data organization, that can really allow you to do more sophisticated analyses. And then just lastly, I just want to really emphasize, and I'll bring this up again at the end, this fact that I really believe strongly that, if you use open source, open data, open publications, open access kind of concept really opens opportunities. It really allows more people to see your research, and I really encourage people to follow sort of, open access approaches whenever they can. Now, I do get limited. There's sometimes I'm in collaborations where I don't have the opportunity to do an open publication, for example, or be able to have completely open data. But I think if we all really push in that direction, as I'll talk about at the end, I think we can really all benefit in the long run. So in terms of bioinformatics, there's a bunch of tools I've developed, and I'm not going to go over all these, but I just wanted to highlight, just for reference, that we've got some tools we've developed for looking at the host, some for looking at the pathogen, including pSort-B for predicting cell surface and secreted proteins, for example, and genome databases that are genera specific, like Pseudomonas Genome Database. And but I will go through a selection of these now, starting with the host. And the reason I wanted to do that is I wanted to really emphasize the need for data integration. We're really at that point now in bioinformatics where we have a lot of data being generated, and we really need to get organized. And we need to get this organized because, examining something as a system, not just its parts, can really be a benefit, as sort of illustrated by this classic story of the blind man and the elephant, and they all feel the elephant, and they feel different parts, and only through everybody talking together about what parts they feel, they figure out that it's an elephant. And so we need integrated data, and we also need ways to view and analyze this integrated data. And so data visualization is becoming a huge issue in bioinformatics as well. But, stepping back from, data visualization is really fun, coming up with cool, artistic ways to show things. But there's some stuff that's really difficult and a pain to deal with, and that is data standards. And I can't emphasize enough how important that is. I like this slide from Paul Fleck about from the EBI about how many ways you can say female in a database. Here's, and you can see there's a whole bunch of things. I love worker bee. That's a great name for what the heck is that show? I don't know how that shows up in a query for things that involve female, but I'll have to find out. But basically, this is an example of a problem that just on a one scale, but this is very common when you're dealing with data. Right now we've got lots of genomes being developed where we're using not the same terminology, and that can really cause a problem for computers, of course, which will not know that female child or female other is actually both female, unless you tell it to. You do the proper standardization. So the other challenge is really that the data integration needs metadata standards. So we don't just need standardized terms, we need standardized metadata, in other words, data about data that categorizes the data and organizes the data more. I mean, we've been doing this for a long time. I mean, card catalogs, for those of you who are as old as me, you might remember card catalogs where we used to have everything grouped into different subjects. And this kind of grouping can be very powerful. And also classifying research data logically. So for example, A interacts with B if C is present, and having statistical associations. These are the kinds of things that are somewhat boring to do. It involves a lot of curation, a lot of reviewing data. It's not, you know, really fun, but it is so important and can be so powerful if done properly. So the other thing I think that is a big challenge in bioinformatics in general is data integration quality. You know, you really have to make sure you're doing quality integration, or you can end up with some real garbage. You can have garbage data and the perfect model, but you'll get garbage results. And you can have perfect data, but you can have a garbage model and you'll get garbage results. So, you know, the holy grail really is basically having that perfect data and perfect model, but the devil's in the details. And really, in the end, what you have to do is spend the time to robustly evaluate and ensure a certain level of accuracy in your data and your analyses. And appreciate that all analyses, you know, the large genomics analyses or the small lab study, all of these are really clues for uncovering the truth. And so, you know, having ways to integrate those together can be very powerful. So one example of sort of integration for us was this InnateDB database, which we developed for two large projects where we were doing systems-based analyses of immune responses. We were looking at mouse and human clinical data sets, modulating immune responses with peptides that were being examined as therapeutics. And the idea was to gain insight into host response, therapeutic mechanisms, identify common pathways, networks, transcriptional regulation. And so to do this, what we did is we made a database of all the known human and mouse experimentally verified gene, protein, RNA interactions and pathways from the major interaction databases. And then we did this exhaustive curation. And, you know, curation is just, it's not trivial, it's very tedious, but again, it's so powerful. So we've now got contextual data of A interacts with B in this cell type under this condition of over 25,000 interactions curated from over 5,000 publications to date, largely thanks to a few curators, and I have to do a shout out for Raymond Lowe, who's been phenomenal, who is all the curation that he's done in my lab. But basically, the concept is that you by putting this data together, by having this data that we gather from the literature, which is very rich data, plus some integrated bioinformatics visualization tools, you can have this sort of user-friendly analysis platform to allow biologists to sort of interrogate their data in a more molecular network context, okay? So the advantage of that is we found this was happening over and over again, of this kind of data integration allowed us to identify key players in a gene expression response that wasn't otherwise present. So for example, here's differentially expressed genes in typhoid patients at days 1 to 3 of hospital admission versus healthy controls. And basically what we found is if you look at this list, you can look at this list of genes that are differentially expressed, or you can overlay them on this network of proteins, and you basically see this cluster here, and essentially you find that there's this key regulator, HNF4A, of the molecular response that itself is not differentially expressed, so it doesn't even show up in the list. It's basically modified in another way, but the fact is that it is actually a key regulator, and this is something that only can be seen if you look at things in a molecular network kind of approach. So we found this kind of pattern very common, and we keep looking at data sets, looking at them as a network, and then start to see these clusters. So we've also been able to, oh, there's some little problem with the title here, but we've also been able to aid some fairly practical analyses. I'm going to highlight two I'm particularly proud of. One is we aided clinical tool development for identification of infection in neonates. That's like the first 4 weeks of life. There's a real problem with neonates when they get infections, that they can have true cases of infection, but they're blood culture negative, and they can really have problems, of course, if they get antibiotics, because that's affecting both their good microbes or their natural flora, as well as this infection, and can really muck them up. So the idea was to integrate this immune and metabolic pathways. By integrating both immune and metabolic pathways, we were able to infer a 52-gene classifier that predicted neonatal bacterial infection and aided identification of these cases that are blood culture negative but still actually an infection, because you're starting to see this immune system change occurring. And then it allowed us literally can potentially allow reduction of antibiotic use in these neonates, which has a huge benefit to them later in life, because there's not good stats about use of antibiotics early in life for children. So the other thing analysis I was particularly proud of was this aiding some therapeutic development. And the reason I like this because it was something that really allows us to go a step beyond the traditional approach. Traditionally, we tend to treat infections as something we either get vaccinations for, which are great, very effective. We'll take an antibiotic after we get an infection. That's okay. I mean, definitely I wouldn't stop from giving an antibiotic to my children if I needed to, but I definitely avoid it unless I have to, because it's also affecting a lot of their good microbes in their body as well. And certainly, you know, there's this problem of resistance, right? In the case of severe malaria, you can give an anti-malarial drug, but in the case of cerebral malaria, you have such a crazy inflammatory response, such a crazy innate immune response, which is your first response to infection, your sort of one that happens in hours after infection. That response is so strong that it basically, that's what's killing you, is this crazy immune response that's basically a severe inflammatory disease. And so NateDB was able to aid in identifying this new immune modulator that basically would be adjunctive therapy that you would take with your anti-malarial. You'd also take this immune modulator, which would dampen down this inflammatory response. And I think that's the kind of thing I would like to see happen more in the future in infectious disease research, where we're looking at both host and pathogen at the same time, and in our treatments. But we do need to organize data more, and you know, this is just interactions that have been added to the NateDB sort of curated interactions over a 2-year period. But there is a lot of data out there in the literature that really needs to get into databases in organized fashion. Unfortunately, it's very difficult to fund projects like that, so I tend to sort of have to do it on the side. But I think it is a really important thing that we have to keep on top of if we're going to actually be able to do sophisticated analyses in the future. But I wanted to go further than this in terms of data integration, and so one project I've really gotten excited about is this IRIDA project, which stands for Integrated Rapid Infectious Disease Analysis. And this is a platform, an informatics platform that we're developing to support real-time infectious disease outbreak investigations. And the idea is to go even further. You know, you're integrating genomics data, epidemiology data, clinical data, lab data, all together in a platform to basically help when you have an infectious disease outbreak. You'd sequence these genomes and you'd integrate this data to try to learn more about origins, with the idea that a genome is basically the ultimate high-resolution molecular typing or fingerprint. And so you can sort of track the spread of this infectious disease by literally tracking this fingerprint, similar to how you might track using more traditional methods. But this is not a trivial thing, but we have had some challenges. And I think one of the challenges is, you know, how do you do this in terms of you've got, say, an outbreak that occurs, say it's in an industry, a food production facility. You don't want to you want to have open data release, but you don't want to just release all this data about this company having some potential microbes that are implying that they have some sort of infectious disease outbreak associated with their food until you actually confirm for sure that is that food. Because sometimes, like that salad situation, you might have associations of foods together, and there might be some, it might not be very clear. So it's often, what we want to do is have a platform that allows public health agencies to do sort of rich genomic epidemiology analysis within the public health agency, where they can keep the data secure and avoid privacy issues. There's issues like, for example, if you say that you've got some food isolates that are associated with certain provinces on the East Coast. Well, you know, there's not that many abattoirs on the East Coast, so you can actually figure out what food processing location this is. So there's privacy issues. So the idea is to encourage that rich data analysis, but also support rapid open genome data release, but with just some minimal metadata to start with. And what we're hoping to do is really complement some other resources that are being developed in the US and elsewhere, where the concept is that you would have this genome sequence data come out as soon as you have this outbreak happening in real time, but it would say something like this isolate is from Canada with this isolation date, with not a lot of identifying information, but allows you to identify clusters of isolates and identify when an outbreak may be occurring, and also analyze those genomes, both academics and public health workers alike. And so the idea is we're making a platform that can basically handle both of these things and be open source, standards compliant. And this is being done with a whole collection of people. In microbiology world, we might say TNTC in terms of all the people involved, or too numerous to count. But basically, a key message here is this is a partnership of you know, myself as an academic, and Will Sow, you know, who kindly introduced me, who's leading the project from the provincial public health agency, and then we've got the national public health agency, National Microbiology Labs, with Gary von Domsler, and basically as PIs, we are together trying to make this resource that will basically work in public health labs at the national level, provincial level, and also have an academic version for academic release. So the idea, again, is this concept that if you can identify through early reporting and through early identification of these clusters, you can have a more rapid response, and you can potentially really cut this natural curve off of an outbreak and cases for an outbreak off sooner, and basically prevent cases and more international spread. And so to do this, really what we need is routine survey sequencing, and this is going to be a very important development in infectious disease control worldwide. This just shows some flight paths across North America because outbreaks tend to follow flight paths more closely than simple geographic distance. And but it's just that, as you can see visually here, I mean, really you don't see a border between Canada and the US, right? It's pretty obvious that we have a very close relationship with the US, so it's very important we develop ways to share this data across countries, across jurisdictions, so that we can track these outbreaks very effectively. So what's been interesting is this IRIDA project was a bioinformatics project. This is a Genome Canada funded bioinformatics project, but our biggest challenge is not the bioinformatics. And I wanted to bring this up because really the biggest challenge has been data sharing agreements, and data sharing even just to be able to view different kinds of data types to enable ontology development or sort of standardized terminology. So I really encourage bioinformaticists, whenever they're involved in projects, not only should you do some obvious things like understand the study design and make sure that it makes sense, that it can be analyzed effectively with the bioinformatics methods at hand, but get data sharing, any data sharing arranged in your project early, so you don't get stuck with being held back by that. So certainly that's a big challenge, and I think it's a big challenge in general in genomic-oriented bioinformatics, where we really need to get some data sharing agreements more in place, so that because really the problem and the limitation is not the bioinformatics for doing a lot of these analyses. So Again, you know, dealing with data standards and curation is not the most exciting thing, but very necessary. So we have been working on addressing some ontology gaps. And for those who don't know, ontologies basically are describe types of entities and relationships between them. So they're not just definitions of what something is, but just how those definitions relate to each other. So basically, we're building on some existing ontologies and then filling in some gaps, for example, antimicrobial resistance information with Andrew MacArthur at McMaster University. So, so we've got all that, you know, base stuff, and this is all stuff that's really necessary, highly important, but then there's the fun stuff, I think anyways, is developing visualization tools. So we've started to do some of that. One example is this island viewer tool, which uses this genome D3 plot, which is JavaScript using a D3 library. It's basically a very sort of elegant tool developed by Matthew Laird in our group, who is, it's basically geared for microbiology data, but could actually be used for eukaryotic data. But the idea was just having a very flexible, dynamic interface for basically looking at data where you can sort of click on regions of this genome and it'll show the corresponding region, the vertical or the horizontal, and you can use your scrolling wheel to zoom in and out here, or you can interrogate this area and it'll show dynamically those same regions in all those areas. And then you can look at multiple genomes as well. And that's where we're actually really heading now, is the big challenge of how do we handle thousands of genomes and potentially hundreds of thousands of genomes. You know, we have some tools that we've developed, like Sniffle. I really like that name, actually. So it stands for Single Nucleotide Variant Phylogenetic Analysis, right? So when you have an outbreak, you can do your SNP analysis or SNV analysis. But when we're doing outbreaks, it makes sense to be Sniffle, right? So we've got a little Sniffle plush microbe doll that goes with this tool. But basically, the idea is with Sniffle and integrating geographic data with work led by Rob Biko and Genghis, the idea is we can start to look at outbreaks in larger scale, but we still have issues around annotating that information with more metadata that is, of course, all organized, but and coming up with better visualizations for comparisons of genomes. So that's a big direction right now that I think many people in genomic epidemiology need to tackle. But we're not even stopping there. I have to say metagenomics is becoming increasingly important, and we've been doing a lot of metagenomics analyses lately because really metagenomics, where you're actually sequencing all the DNA in a sample, allows you to see all the microbes that are there or have been there, and you basically can really identify everything, even if they're not culturable, and you can track, you know, what's happening in environment, be it in your gut or in the water. And so we have one project where we're looking at clean and fecally contaminated water, looking upstream and downstream of a pollution site in multiple sites affected by agricultural pollution and urban pollution, and we're trying to track, you know, how these microbiomes change and how they change over time, over the course of a year, and how they change over space, and identifying markers of water quality. Because as you guys may know, often your beaches get closed due to high coliform counts, but coliform count is a horribly inaccurate measure of water quality. Basically, all coliforms are not pathogens, and not all pathogens are coliforms. So basically, you have a problem with false positives and false negatives. So I'm not going to talk about this project because of time, but I just want to say that certainly we've been really enjoying integrating that, and I think a key thing with projects like IRIDA, where we're integrating outbreak analysis, is we have to integrate metagenomics data as well. I will give you one little teeny tiny analysis we did of microbiome analysis at a proposed northern Canadian mine site, just to show you that there's a lot of environmental applications. So in this case, we had a watercourse that had a natural decline in copper downstream of where the copper was introduced, as shown here in this graph of the copper concentration in the water. And so you get this like really low pH when you introduce copper to water. You know, it kills a lot of stuff. And so we obviously don't want to downstream of a copper mine and have a lot of copper being introduced. But what was interesting at this natural site was there seemed to be this reduction in the copper at a level that could not be explained by dilution of the adjoining streams. It could not be explained by physical mechanisms. So they wondered, this company wondered if, mining company wondered if there was actually some microbes there that were being involved in some sort of naturally occurring remediation. So we went in and did some sampling of microbes in that environment, looking at these different sites upstream and downstream of the copper introduction. And the key message here is the minute you get that copper introduced, you do have a large increase of this one species that I can't release because we're literally sent the paper out to review with this company, and this is an example of open data. I would love to say everything, but I've got to just make sure this company is okay with us releasing what that species is. But basically, we've got this species here that is clearly, maybe I should say we can detect it more at the general level, that is clearly taking over and very abundant in that location. And most notably, it is not known to transform copper, but its close relatives of species are known to be involved in transforming iron and other metals that make it sort of interesting in terms of its potential to potentially transform copper. So obviously a natural thing we need to do is we can use our metagenomics data to figure out what its gene content in is, the closest relatives, and knowing how they get cultured, which we do know, we can try to culture this microbe and see if it actually could be used for remediation. And in particular, I'm excited about this because this is a nice concept of going in with microbiome analyses and identifying naturally occurring remediation to help do remediation of any kind of disruption humans make using native microbes at a site, okay? So anyways, I do have to mention a couple of cautionary tales that just, we have a paper that we're just under revision right now that really shows that metagenomics software accuracy varies greatly, and it's really important that you be very careful when you do metagenomics analyses. Always have controls of a sort of blank water control and spiked DNA control to check that your methods are performing appropriately and that your data is okay and your sequence is okay. You can get cases of contamination that will show up if you're not careful. So just always remember, you know, metagenomics, like any kind of experiment, you need those controls, and you need to make sure that your metagenomic software is producing the results that you should see with those controls. If you have 8 species you're putting into a control, your metagenomic software, you should figure out the settings for getting those 8 species back. So I have to say, though, you know, why care about microbes? This is my sort of attempt at a summary. Really, I can't emphasize enough, they're very cool. And so why you should care about microbes as a bioinformatist is some things have nothing to do with bioinformatics. They do so much. They're very cool microorganisms that they impact us. They're playing roles. We're finding out they play roles in so many things we didn't appreciate, like jet lag, also in chronic disease. And they do so much for the world, literally. So, you know, they are very important things to care about in terms of life on Earth, but also we're learning so much about them. It's been a really fun journey lately with microbiome research, where we're starting to do microbiome analysis of our guts. We're doing microbiome analyses in homes, around the Earth. There's the Kitty Biome project is really awesome, where they're looking at microbes in kitties. And basically, we've got this concept of this whole new world opening of understanding how these microbes are changing in our environment and the role they play, and using them as markers for various things, including for chronic disease, that mean that really, as a researcher, regardless of whether you're doing microbiology or not, you should pay attention to microbes, because they can become important markers in different diseases being looked at or different environmental conditions being examined. And also, they're really a bioinformatics goldmine. I mean, they're interesting data analyses, visualization challenges that we're facing, because we're often in microbial informatics, we're tending to deal sometimes with some of the challenges that the human genome projects, will face later as they sort of ramp up to that sheer number of genomes. And I would also argue that there's just so much to integrate. There's so many interesting discoveries that it really is a gold mine in terms of discoveries to be made. So in short, why I think you should care about microbes is not only because they do so much, but also there's very similar challenges to non-microbe research. And so you can apply a lot of the ideas of ways that people are handling the things we're doing for sharing data, the ways we're doing integrating clinical and epi and laboratory data, the way we're handling visualizing large data sets of genomes and overlapping metadata. A lot of these things could be applied to, your favorite organism if it's not a microbe. And I'm actually realizing that's also an acronym for your fly is open, so that's probably not the best acronym to use. But anyways, the point is that hopefully I've shown you that microbes really are important, and that they are something, and I encourage you to keep on top of microbial informatics research. Okay, in closing though, I just want to address one issue, and that is bioinformatics stress. So a lot of bioinformaticists are going, because there's so much data out there, right? It's just crazy, and there's more data coming, and it's like, oh, are we going to be able to handle this? And so I wanted to mention, there's a lot of different things you can do for stress. Eat well, exercise, just remember those basics, right? But, let's work together more. I mean, there's nothing like getting together with people to de-stress you when you start to realize everybody's dealing with the same things. And if you start to deal with things together as a group, it can become a lot less stressful. And so I would really love to see more integration, data integration via computers and data integration via people. And it's really all about communication. We shouldn't shy from sharing. I think it really comes down to open access is important. And for computers, ontologies and data standards are really key for basically doing data integration. For humans, it's more about getting and keeping their attention if you want to integrate them. So like you can have paper carrots for encouraging them to participate in a project. That's what we've had a lot of success with, where we say, if you help us with this project as a big community annotation project, for example, you will get an authorship on this paper, and that's worked really well. I have to say the other way to get their attention is beer, and I kid you not, I think one of the best ways you can integrate and bring people together is just through pizza and beer. And so hence, we have this van bug concept of where we have always pizza afterwards, and I really want to encourage that concept of bioinformatics community integration. I think we really do have to have mechanisms to bring that together. And because of that, I really want to thank VanBug, fantastic seminar series, and fantastic team of organizers, including the present organizers. Thank you so much for all you do to keep this running. And I also have to do two more thank yous. One is thank you for any feedback anybody's given for this draft bioinformatics computational biology strategic framework. If you don't know about it, please go to this URL and give your feedback about what you think we should be doing in bioinformatics for Canada. It's due by September 25th, so please complete that survey. And I just have to say thanks to my kids, because this is going to go on YouTube, and I want them to see on YouTube me saying thank you, because they are so awesome, and they really are my inspiration. They're fantastic. But also, last but not least, I lied. There's actually three things to thank, but because absolutely, first and foremost is my fantastic lab members who really make it all possible, and I want to thank them for all their fantastic work. With that, I will be happy to take any questions, and thank you.
Yep, that was great. Thank you. I was really interested in the story about the copper, that when you went farther downstream to the C and D sites, the communities looked totally different. So do you know if that was, were those other species kind of there before and just pushed down, or is it something about the sites that they just had different kinds of communities to start with?
Yeah, this is very typical of microbiome analyses right now. We're like a kid at a candy store when we don't know what we're doing and we're just like, wow, this is so cool, but you don't really understand how that candy is getting there. So we basically are seeing these things, but we don't really know what's going on, and we basically need more in-depth study. But I can say for sure it's interesting to me that we do see some species coming back that were there before, which is interesting. But there's absolutely no doubt about it, there is a change occurring. I think there's two things that are notable about that. One is that this is naturally occurring, so maybe we shouldn't fear so much, that if some mining company comes in and they're causing some disruption, if we can say, for example, put in a little and if it turns out that this microbe is responsible for doing some sort of transformation of copper, we can use a little bioreactor, just downstream of this mining site, and at least keep on top of the level of copper being produced, and hopefully we can maintain this. And what's nice is now we've got this baseline data. We know what's naturally occurring. But absolutely, like you say, these are great questions, and I think we have to investigate it further. Yeah.
I was just wondering what is exactly keeping you from releasing the name, or is this work entirely tied to the concept of the company, or can you conduct it independently?
This is actually work that is driven by an environmental consulting company and British Columbia funded project. So it's really about usually when you have an industry interested in doing something in an environment, you have to have environmental consulting that occurs. And so these environmental consulting companies that have to do an assessment. And so this is really coming from that assessment background of trying to come up with a better assessment of the situation, because usually you're always wanting to go in, you might do a disruption, and then you want to put it back into the same state as much as you can as it was in the first place. And so this is really, the main message here is more just about that I think we can use microbiome analyses in a lot of different ways and include ways that could help us understand more what's happening in different environments and be able to sort of have that key data about how things are changing in environments. Yeah.
Is there a kind of a food web, like any given microbiome? So, like, are there like competitors with very smaller?
You mean microbes eating microbes kind of concept? That's a very good point. So, there is a study I did way back when of a protist that basically had an unusual gene content, and then it had a bacterial gene in it that was very, very highly similar to other bacterial genes, such that it looked like it was horizontal gene transfer. And the mechanism we were thinking for how this really rare case of bacteria eukaryotic horizontal gene transfer is extremely rare. And we think this might have happened because literally these protists are eating bacteria. And so there's certainly on that level, there's sort of microbes eating microbes, absolutely. But in terms of a hierarchy, that's a very interesting point. I certainly don't know much about that, but I can say that we are just really starting to understand just the level of dynamic change occurring, let alone understanding the relationship between microbes. We're just trying to understand what's there and how they're changing. So, for example, we went into this watershed project, and we didn't know if we could find markers of water quality, because we didn't know if the microbiome would be changing every day or every hour or every month. It turns out that over periods of hours, it doesn't change very much. But over seasons, it really changes. So there's definitely a seasonality. In fact, it's a dry, wet season, just a two-season situation. As you know, in BC, it's sort of like dry or wet, right? But basically, we're literally at that stage of just trying to find out what's there. And then we have to go to that next step, right, of relationships between organisms. Yeah.
Just one question. I don't understand something that would add or use on the
Yeah, great question. So there's no sort of even bad microbes, quote, bad microbes aren't really bad, and that, you know, they don't, there's no microbe that really kills 100% of people, for example, or a host. You know, any given microbe is pathogenic to some organisms, but not to others, you know. And any given microbe can be beneficial to some organisms and not to others. So it's very contextual. You know, what is disease is really a question of, you know, what the host is, what their genetics are, and their current environment, what their you know, what the pathogen is and what environment is involved. And so that's why I'm really interested in that concept of really integrating things more so that we can sort of start to say, okay, we've got this pathogen involved in this outbreak. There's really cool publication that just came out where they're doing human microbiome sequencing, and the contaminating human DNA is being sequenced, and they get actually genetic information about the host. They can actually get the microbiome and host together, and it's going to be sort of funny because it's, you know, we've always traditionally thought of human studies where we've got this contaminating microbial data. Now it's becoming like humans are the contamination, and we really care about the microbes, right? So basically, this concept of being able to look at the microbiome and the host genetics, and then also looking at the epidemiology of a situation. You know, most infectious disease outbreaks are, you know, not caused by some sudden increase in virulence. They're often caused by some sort of change. You know, like there was a measles outbreak that occurred here in BC, but that was because of you know, the Olympics, I guess it was, right, Will? That basically, you know, brought a bunch of people together in close quarters, and we ended up having a measles outbreak as a result.
In terms of data integration, I guess we sort of have two models that you can use. We have one where data is out there and it's curated, and then you use that curation to bring the data, to integrate the data. Then you have another model where you are creating the data and integrating it at the same time. And I guess I wonder how you see those two approaches, whether one is better, or whether you think in the future whether there will be a responsibility of creating data to make sure to integrate it, or whether it's always going to be third party after the fact.
Great question. I know it's hard to say what's going to happen. I have to say, if there's one thing I've learned is that researchers, people who generate data are notoriously not very good at curating that data. You know, they really get very excited about getting the data, and then they want to analyze it the way they want to analyze it. But it's like pulling teeth getting people to add extra information about a genome and put that in, for example, in CBI in a very organized format. So that's why we've had to do a lot of this curation, right? Is because people don't want to take, you know, when they publish their paper, they could, you know, technically everybody could get everybody to, like, you have to submit that information to an ATV whenever you publish a paper about an immunity. But I mean, when we, with the Pseudomonas database, database tried to have a web submission form for if you wanted to submit an update to a gene annotation, people weren't using it. They would just email me, or email the curator, the coordinator, Jeff Windsor, fantastic person who's been coordinating this for a decade, and they would just want to email the information, just something that was just easy, right? So what we learned from that, which is really important, is you want to get that information from people, but you do want to have these people that have sort of informatics background and ontology background or curation background who are going to organize the data well. And so that would argue for the model of, that people are not going to be so good at doing that, and that we should have these people doing that. but it's really, really hard to fund that. So, you know, I think we have to do a bit of a mixed bag of really putting the pressure on people to appreciate the need for this kind of data organization. And I think actually that starts at the, you know, we should be at the undergrad level, getting people, you know, mold minds, get people to realize the importance of annotating your information properly and getting all that curation done. If you just do it on a little project, it's much easier, right? Okay. Yeah. I think that'll be the last, or yeah, question. Yeah.
So you're talking about how epidemics are kind of very closely related to flight paths. Based on past data, is there like any predictive models about how epidemics will spread in the future?
There is some really nice papers that have come out in the last few years on just, you know, how you can, basically knowing these flight paths, how you can predict how an infectious disease will spread. There is some nice modeling also done of Ebola recently. Yeah, absolutely, that's very valuable, and they've got great stats on, you know, the best cities to live in where you're likely to not get very, you know, infection versus other cities that are like, you know, hotbeds. And I don't know, I just think of zombie outbreaks, and like, I'd go to the northern Canada, I guess is where I would go. But basically, yeah, and so this is a very powerful area. And one thing, you know, I really like to encourage people to do is to remember that power of integrating all these different ideas, right? You know, there's a lot of great integration if you start integrating ideas from social science regarding you know, behavior with mathematical modeling, you know, with bioinformatics, with genomics. I mean, there's some really cool things we can do, and so I hope some of you students will take that in mind and come up with some great innovative ideas. With that, I think we're done.